Assalamu alaikum and good morning, everyone. I'm Rabia Zara, PhD scholar from Government College University, Faisalabad. Welcome you all to 4th International Conference on Applied Zoology 2021 on the behalf of the organizing committee ICAZ-2021. I'm thankful to the committee for providing me this opportunity to communicate as a moderator of second session of day 1 of ICAZ 2021. Let's begin our session with the first talk of Dr. Mina Amdi from Agriculture Research, Education and Extension, Iran. She will deliver her talk on fish waste processing plant for compost production. Please, Dr. Mina Amdi.
In the name of God, my name is Mina Ahmadi. I'm a researcher at National Fish Processing Technology Research Center from Iran. The subject of my article is about fish waste, fish processing plant for compost production. Fish processing waste also have a high potential for compost production due to their richness in organic compounds. Fish waste is used as organic fertilizer, nutrients for agricultural purposes or as a renewable source. Fish waste has been proven to be an organic fertilizer and nutrient for both agricultural uses and soil strengthening is useful. Composting is a biological process that takes place under aerobic conditions in the presence of oxygen. The final pH of the compost is strongly influenced by the organic matter, the decomposition states of the compost, and the increase of the modifiers. A large amount of salt in compost affects seed germination and root health. Compost produced using fish waste and by examining the electrical conductively pH and carbon nitrogen ratio can be used as organic fertilizer. Disposal of fish waste causes environmental problems and the disposal process is also problematic. New technologies must be a way to use and improve this waste and turn it into organic matter, provide usable before disposal and to reduce contamination. Fish waste is biodegradable organic waste. The waste generated through processing of fish, crab, and shrimp amounted to 30 to 60%, 75 to 85%, 4 to 80% respectively. The selection of fish waste as raw material composting is not without resource. This is due to fish, including food that is easily decomposed, so that the remaining ingredients in the form of fish head, fish meat fragments, fish entrails, fish fins and tails, if not handled carefully, will cause a strong stench. The stench will invite many flies and can cause various diseases. The importance of fish compost, it is economical, fairly odorless, biological beneficial practice for seafood operations. Composting resolves the disposal problem and produce a valuable product, a humus-like material that has several marketable use from soil conditioner to horticultural grow medium. It decreases the potential for surface and groundwater contamination. Composting destroys disease-causing organisms and fly larvae. Compared to other disposal options, composting is not a costly method of fish waste disposal. Composting result open and in the indigenous population of microorganisms from the environment and is carried out with the help of many organic materials fairly odorless. Microorganisms such as bacteria, fungi, and actinomycetes account for most of the decomposition that takes place in a compost pile. The amount of waste, byproduct, and implicit fishing depends on several transient factors, including fish species, size, season, method of fishing, processing method, local regulations, and market. There are various factors that affect the composting processes and determine the level biological activities. The main factors are moisture, pH, carbon-nitrogen ratio, oxygen, and temperature. When composting such waste as fish parts, the fish waste is mixed with the plant waste like wood chips, leaves, bark, branches, peat, or even sawdust. As microorganisms break the fish down, they generate lots of heat, which serves to pasteurize the resulting fish compost, in turn eliminating any odor and killing disease organisms and with seeds. After several months, the resultant product is rich humus loaded as a nutrient, wealthy fertilizer for soil amendment. Composting fish does not need to be a complex operation. The basic requirements for composition of composting fish are a source of carbon and nitrogen, which is where the fish scraps come into play. A simple recipe is 3 parts carbon to 1 part nitrogen. Other integral factors for composting fish are water and air, about 60% water to 20% oxygen, so aeration is necessary. A pH of 6 to 8.5 is needed. A temperature of 130 to 150 degrees Fahrenheit during the decomposition process, at least 130 degrees Fahrenheit for these successive days to kill any pathogens. The compost pile will cool to the ambient temperature after several weeks, and when this occurs, the compost is ready. Before constructing a fish waste composting facility, location of the site, management of surface runoff, leachate, moisture, odor, and dust should be considered. The main cause of odor from fish waste composting facilities is anaerobic condition within compost heaps. Under anaerobic conditions, the main gas generated from composting is odorless carbon dioxide. Under anaerobic condition, conditions, odors, gases, organic sulfides, and volatile fatty acids are produced. Odor management therefore requires that aerobic conditions be maintained by regularly turning the compost heap or using ventilation structures. Odor can be further reduced by maintaining an appropriate ratio of carbon to nitrogen in the compost heap. This means using an appropriate amount of sawdust relative to the amount of fish processing waste, and is discussed later in this document. In the end, it can be said as a result that animal byproducts of the fish industry, half-dead fish, are products derived from the processing of fish or consumption. Proper purposes can be applied in agriculture and gardening as fertilizer. Upon having first been subjected to proper processing, they can be composted and used along with other organic materials. Thanks for your attention.
Thank you, Dr. Mina Amdi, for such an interesting talk. Now I would like to call Dr. Samreen Gul Khan from Government College University, Faisalabad. She will deliver her talk on synthesis, antibacterial and hemolytic activity of N,4,4-morphonenyl phenyl to 5-aryl aracyl substituted 1,3,4-oxidizole 2-vial thioacetamides. Please, Dr. Samreen.
Everyone. I'm Dr. Samreen Khan and currently working as an assistant professor at Department of Chemistry, GC University, Faisalabad. I would like to thank the organizing committee of ICAZ-2021 for providing me this opportunity to share my work at 4th International Conference on Applied Zoology 2021. So today the topic of my presentation is synthesis, antibacterial and hemolytic activity of N,4,4-morphonenyl phenyl 2,5-disubstituted 1,3,4-oxidizole 2-thioacetamides. In my topic, I have mentioned two heterocycles. One is morpholine. It is a 6-membered heterocyclic ring containing two functional groups. One is ether and the other one is amine. Morpholine is found to have very good biological activities, like it is part of different already available medicines in market, like linezolid, it is an antibiotic drug, gefitinib, it is an anti-cancer drug, dextromoramide, and an analgesic. Morpholine is found its application in agriculture as a fruit coating as well as a fungicide. The other heterocycle was oxidizole that I mentioned in my topic. Oxidizole is a 5-membered heterocyclic ring containing 2 nitrogen atoms and 1 oxygen atom. Depending upon the position of these 2 nitrogen atoms, this oxidizole has its 4 isomers: 1,2,3-oxidizole, 1,2,4, 1,2,5, and 1,3,4. Depending upon the position of this 2 nitrogen atoms, this 1,3,4-oxidizole have been reported to have significant activities like anti-inflammatory, anti-enzyme, antiviral, antifungal, anti-cancer, antioxidant, etc. Here are some drugs available in market having oxidizole moiety common in them, like raltegravir, it is an antiretroviral drug, zibotentan, an anti-cancer agent, oxadiazone, a herbicide, and metoxadiazone, a pesticide. Pakistan is a developing country and drugs are in a process of development. So the main intention of my work was to design and synthesize a novel series of polyfunctional molecules having morpholine, oxidizole, and acetamide moieties together in one skeleton. It was also speculated that these moieties together in one skeleton will boost up the biological activity of molecule. Here is overall plan of my work. My work consists of 3 steps. First was synthetic, the second was characterization, and the third one was biological screening. For structural elucidation, I have used these 3 techniques. Here is overall scheme of my work. I took different organic acids and they were converted into their respective oxidazoles through different steps. And then in this way, I prepared a series of different oxidazoles, and I have chosen these 4, 1, 2, 3, 4 morpholine bearing different compounds. And then when they were reacted with oxidazoles, they gave their different substituted respective polyfunctional molecules having oxidazole as well as morpholine moieties together in one skeleton. In scheme 2, I choose 4,4-morpholinide phenylacetamide. For scheme 3, I have chosen 2,4-morpholinide. Here at this position, it is at position 2, 2,4-morpholinide phenylacetamides. For next scheme, I have chosen the 2,5 substituted 1,3,4, 4,4-morpholinide phenylacetamide. And then I have chosen this simple morpholine. And for last scheme, I selected 2,thioethyl morpholine. Here is scheme for synthesis of oxidazole from its respective acid. Acid was reacted with sulfuric acid in the presence of ethanol, and then they were converted into their respective ester. Then ester were converted into their respective hydroxides in the presence of hydrazine hydrate, and hydroxide were finally cyclized into their respective oxidazoles by reacting with potassium hydroxide and carbon disulfide. I have mentioned all the reaction conditions and reagents here. Here are different 5 substituted aryl or alkyl group of 1,4-oxidazole 2-thiols. Then I choose 4,4-morpholinide benzene, and it was reacted with bromoacetyl bromide to give an electrophile that is 2-bromo and 4,4-morpholinide phenylacetamide in the presence of sodium carbonate. Is the proton NMR of the electrophile. In the aromatic region, we can see as these 2 protons have same environment, so they give a doublet at 7.31, while these 2 protons that are adjacent to morpholine, they give a doublet at 6.76. While signal for these methylene groups appear in the aliphatic region. Here you can see at 3.8 and 3.7. I have merged actually 2 different NMR spectra in the aliphatic region and in the aromatic region. Then the electrophile was reacted with series of 5 substituted 1,4-oxidazole 2-thiol to give its respective 4,4-morpholinide phenyl 2,5 disubstituted 1,4-oxidazole acetamides. One of the compound is here. It was obtained as white amorphous solid, having melting point from 224 to 226 degrees Celsius. Here is its IR. It's the NMR of the compound. Is the mass fragmentation data of the compound. In the mass spectrum, you can see molecular ion peak at 410, while other fragment ion peaks appear at different values. Base peak was appeared at 204. So this mass spectra confirm the formation of our final derivative. Here is proton NMR of the another compound. In the aliphatic region, we can see clear peaks. Peak for this methylene appeared at 4.3. While signal peaks for these protons appear as a multiplet. For this proton appear as a triplet. In the aromatic region, signal for these two protons appear as a doublet at 7.88, while signal for these two protons appear at 7.12. Signal for this proton attached with nitrogen is highly deshielded. It appears at a value 9.55. Then the next reagent was morpholine. It was reacted with 4-bromomethyl-benzene sulfonate chloride to give 4,4-bromomethyl-phenyl sulfonate morpholine in the presence of sodium carbonate. Is the proton NMR of the electrophile. In the aromatic region, signal appear as a doublet for this proton, while this proton also shows a doublet. And it's the aliphatic region. In aliphatic region, signal for methylene groups present in morpholine appear as a triplet with different G values. Then this electrophile was reacted with a series of different 1,3,4-oxidizole to give its respective 5 substituted 1,3,4-oxidizole to 4-morpholenyl sulfonyl benzyl sulfite. One of the compound was obtained as off-white amorphous solid in 78% yield, having melting point 162 to 164 degrees Celsius. Is the proton NMR of the compound. So from all spectral elucidation, it was confirmed the synthesis of single and pure derivatives. After successful characterization, compound were also checked for their antimicrobial activity against different bacterial as well as fungal strains, and compound 6D and 6I were found to have very good antibacterial activity as well as antifungal activity. Compound were also checked for their hemolytic activity using tritone as a standard, and compound 6H was found to have minimum cytotoxicity. So we conclude that we have prepared a novel series of different oxidizole derivatives having morpholine as well as oxidizole moieties together in one skeleton, and compound 6D and 6L were remain the best inhibitor against all selected bacterial as well as fungal strains. Compound 6H shows very low toxicity relative to trisitol employed as a reference drug. In addition to low toxicity and moderate good thrombolytic activity, the synthesized compound possesses excellent to moderate antibacterial activity relative to ciprofloxacin using as a standard drug. So all compounds could be considered for further clinical studies and might be helpful in synthesis of new drug for treatment of different diseases. Further modification may produce better results. That these candidates may be new drug candidates. Thank you for your attention.
Dr. Sabreen for such a nice presentation. Now I request Mr. Umer Shah from Kohart University of Science and Technology to present his talk on biochemical characterization of multi-drug resistant bacteria in refrigerated fish. Please, Mr. Umer Shah.
Bismillahirrahmanirrahim. My name is Umer Shah. I am MPhil Scholar, Department of Zoology. The research title of my presentation is about biochemical characterization of multi-drug resistant bacteria in refrigerated fish. The research work was done under the supervision of Dr. Farman Dawar and Dr. Mubashir Hussain, Department of Zoology, Cust. Presentation outlines are introduction, study gap, objectives, material methods, results, conclusion, and references. Introduction. Fish are of great importance and play a pivotal role in the economy of a country due to their high nutritive value, containing high protein contents, carbohydrate, and omega-3 fatty acids. Refrigerated fish are exposed to high risk of contamination during long chain of catching, transportation, and freezing from different sources. Majority of the foodborne diseases are caused by Salmonella, Staphylococcus aureus, and Escherichia coli, commonly linked to consumption of infected fish. Consumers have high chances of infection or intoxication after intake of these fishes contaminated fish as a source of food. Water quality also influences quality of fish because water and ice used in processing fish may contaminate the whole processing plant, thus increasing the chances of infection. In aquaculture industry, the use of antimicrobial agent has played an important role to inhibit bacteria. Antibiotics are used in aquaculture to treat bacterial diseases, but the misuse of antibiotics in aquaculture could create public health problems. Antibiotic resistant bacteria cause variety of infection and it is increases at enormous rate globally. Various types of pollution are resistant in bacteria against variety of drug that can enter to human body after injection of these fishes contaminated by these multi-drug resistant bacteria. Various types of pollution are resistant in bacteria against variety of drugs that can enter to human body after injection of these fishes contaminated by these multi-drug resistant bacteria. The study gap of this presentation is in view of the several ways refrigerated fish could be contaminated with various MDR bacteria. There is no previous data about MDR bacteria in refrigerated fish in district cohort. The objective of this study was to isolate and characterize multi-drug resistant bacteria from refrigerated fish of district cohort. The second objective was to determine the antibiotic susceptibility pattern of the potential bacterial species. Now coming toward material method, first of all, ethical approval was obtained from ethical committee cust, and then sample collection of fish were taken, and after collection, dissection of fish and isolation of different parts like scale, gills, intestine, and liver were obtained. After that, isolation of bacteria. After isolation, morphological identification of bacteria, and for further identification, morphological identification of bacteria were done, and at last, antibiotic susceptibility of bacterial species were done. First of all, sample were collected from random fish in main market of district cohort. After collection, dissection of fish were done and isolated 5 different parts. And after isolation, serial dilution method were done for the isolation of bacterial species, and then morphological identification and then biochemical identification were done, and at last antibiotic susceptibility pattern were checked. This is the microscopic appearance of different bacteria: gram-negative bacteria, gram-negative rod, gram-negative long rod, and gram-negative cocci. Biochemical test for the identification of different bacteria. This is the citrate test, SIM test, or catalase test, and oxidase test for different bacterial isolates. Isolated bacteria from refrigerated feed isolated through specific media. This is SS agar, Salmonella Shigella agar, this is Cetramide agar, this is MSA agar, and this is EMB agar. Now coming toward results, biochemical identification of isolated bacteria from refrigerated fish. These are the isolated bacteria: Staphylococcus aureus, E. coli, Pseudomonas, Shigella, and this is gram staining result and its morphological appearance. And then the result of catalase, oxidase, sulfur indole motility test, and citrate test for these bacterial isolates. Isolated bacteria from rapid fit identified through specific media. These are the bacterial isolates, and this is the selective media for different bacterial isolates. This is the result for mannitose salt agar. This is the result for EMB agar. This is the result for citramide agars, and this is the result on Salmonella Shigella agar against these bacterial isolates. Number of bacteria associated with the different infected path of different fish. This is the fish name Lebiorheta, Torpitoria, Serpenius carpio, or Hicromus nicotellus, and Giblion carta. These are the number 6 different number of fishes. These are the infected parts: skin, gills, intestine, liver, and muscles. Associated to the devolution factor 3. Number of colonies are, and this is the associated bacteria from these infected parts. Number of bacteria associated with the different infected part of different fishes. This is the fish number, this is the fish names, infected part, and these are the bacterial species associated from different infected organs. Inhibition zone ratio for tested antibiotic against isolated bacteria. I have selected 12 different type of antibiotics having different mode of action and having different class. First of all, I applied 5 different antibiotics for against these 4 bacterial isolates, and then I applied another 5 antibiotics for these bacterial isolates. And at last, I applied 2 different antibiotics for these bacterial isolate and check its susceptibility and multi-drug resistant pattern. This is the result for this is the table for multi-drug resistant. This is the antibiotic category having different mode of action. This is the isolated bacteria having total number 27, having total number 21, having total number 15, and total number is equal to 19. This is the bacteria. This is the resistant bacteria and its percentage ratio. Antibiotic assay against E. coli and Salmonella species. These are the antibacterial agents having 12 number analytic as ciprofloxacin, sulfametazole, cefiximine, empenin, etc. And this is the susceptibility pattern against these 2 isolate. S shows sensitivity ratio for these antibacterial agent. I shows intermediate ratio for these antibacterial agent. R shows the ratio against resistance. R show resistance against these antibacterial agents. This is the antibiotics assay against Pseudomonas and SRS species. These are the antimicrobial agent, and these are the Pseudomonas and its total number 17, and this is the Staphylococcus species total number 21, and this is the susceptibility ratio. S shows sensitive, I show intermediate, and R show resistant. These are the sensitive isolate. These are the intermediate isolate, and these are the resistant isolate against these antibacterial agents. Now coming to our conclusion, to avoid bacterial infection, fish are treated with antibiotic which lead to develop antibiotic resistance in the normal bacterial flora. Thus, excessive use of antibiotic has reduced the effectiveness of treatment normally used for the control of bacterial infection, and thus cause contamination of soil, water, which leads to the development of new antibiotic resistant bacterial strains. Acknowledgement. I special thank to Dr. Farman Dawal, Assistant Professor, Department of Zoology, CUST, and my co-supervisor, Dr. Mubashir Hussain, Assistant Professor, Department of Zoology, CUST, as well as special thank to all faculty members and lab fellow. This is the references, and I like to special thanks to all my class fellows.
Thank you, Mr. Umair, for such an informative talk. Proceeding towards next, now I request Ms. Mavish Khan from University of Veterinary and Animal Sciences to present her talk on evaluation of dietary vitamin C requirement of hypophthalmitic mollitrix fingerlings and its effects on growth, hematology, and serum enzyme activities. Please, Ms. Mavish Khan.
Welcome to International Conference on Applied Zoology 2021, organized by the Applied Zoological Society of Pakistan and Quaid-e-Azam University, Islamabad. My name is Mehwish Khan. from Department of Fisheries and Aquaculture, University of Veterinary and Animal Sciences, Lahore. My topic is evaluation of dietary vitamin C requirement of hypophthalmic mollitrix fingerlings and its effects on growth, hematology, and serum enzyme activities. Introduction. Vitamins primarily act as cofactor and necessary for the maintenance of normal metabolic processes. Vitamin C is a water-soluble vitamin and acts as an immunomodulator and powerful antioxidant. Most ileots are unable to synthesize vitamin C due to the absence of L-gluconolactone oxidase that is required for vitamin C synthesis. An exogenous source of vitamin C is required for normal growth and physiological functions. Advantages of vitamin C include enhanced immunity, increased enzyme activities, iron metabolism and hematology, vitamin E regeneration, hormone and collagen synthesis, skeletal maintenance, and wound healing. Vitamin C deficiency can cause structural deformities, suppressed immunity, retarded growth, anorexia, anemia, and internal hemorrhaging. Objectives: to determine the effect of vitamin C on growth performance, proximate composition, body indices, and hematological parameters, to determine vitamin C concentrations in the liver, and to analyze the effect of vitamin C on serum biochemistry, antioxidant enzyme activities, and survival rate against aeromonas hydrophila. Experimental site. The research work was carried out in Fish Seed Rearing Unit, Department of Fisheries and Aquaculture, UAS, Ravi Campus, C Block, Patoki. Composition of experimental diets. Ingredients include fish meal, sunflower meal, canola meal, wheat flour, rice polish, fish oil, vitamin premix, that is vitamin C free, mineral mixture, lysine, methionine, monocalcium phosphate, and L-ascorbic acid as a source of vitamin C, which supplemented at the level of 0, 50, 100, 200, 400, and 800 milligram per kilogram in experimental diets. Experimental layout. Diet formulation include mixing of ingredients, dough formation, pellets drying and storing in self-sealing bags. Fish husbandry include acclimatization of fingerlings for 15 days, 25 fish stocks in each replicate. Fish fed with the test diet twice a day. Physicochemical parameters kept constant, and feeding trial was carried out for 60 days. Challenge trial. After 60 days of feeding trial, 10 fish from each treatment were harvested for analysis. However, remaining 15 fish were challenged with aeromonas hydrophila and survival rate was observed after 10 days. Analysis. Analysis include growth performance, body indices, proximate composition, hematology, serum biochemistry, antioxidant enzymes activity, and T-bars assay, vitamin C content in liver, and survival rate against aeromonas hydrophila. Results: growth performance. Final weight and absolute weight gain show a significant increase with increasing vitamin C dose up to 200 milligram per kilogram level. Thereafter, a non-significant effect of vitamin C was observed. Weight gain percent and specific growth rate was significantly increased with increasing vitamin C dose up to 200 milligram per kilogram level and then became rather steady. Feed intake and feed conversion ratio show a dose-dependent improvement with vitamin C. However, survival rate remained non-significant. Proximate composition. Vitamin C supplementation show non-significant effect on whole body moisture content. However, whole body food protein were significantly increased with increasing vitamin C dose up to 200 milligram per kilogram level. And after this, a non-significant decrease was observed. Crude fat or supplemented groups were significantly higher than the control group. However, there were a non-significant difference among the supplemented groups. Vitamin C non-significantly affect the ash content up to 400 milligram per kilogram level. However, a significant decrease was observed at 800 milligram per kilogram. Body indices. Hepatosomatic index show a significant decrease with increasing vitamin C up to 100 milligram per kilogram and then increased. However, viscerosomatic index shows significant increase with increasing vitamin C dose up to 100 milligram per kilogram level and then decrease. Hematology, hemoglobin, white blood cells, and hematocrit show significant increase with vitamin C dose. However, a significant decrease observed at 400 milligram per kilogram level in hemoglobin and 200 milligram per kilogram in hematocrit values. Red blood cells and mean corpuscle hemoglobin concentration were also significantly increased with vitamin C dose. However, maximum values were observed at 200 milligram per kilogram level. Serum biochemistry. Alkaline phosphatase activity significantly increased up to 100 milligram per kilogram level, and then a non-significant decrease was observed. Alanine aminotransferase activity showed significant decrease with increasing vitamin C dose up to 200 milligram per kilogram level, and thereafter a significant increase was observed. Aspartate activity and glucose content were significantly higher in the control group as compared to the supplemented groups. However, supplemented groups remain non-significant from 100 to 800 milligram per kilogram level. Antioxidant enzymes activity and T-bar assay. Superoxide dismutase activity significantly increased and then followed a non-significant consistence and increased and decreased at 800 milligram per kilogram level. Catalase activity shows same pattern, however, it shows significant increased at 800 milligram per kilogram level. GPX and T-bars of supplemented groups were non-significantly different. However, lowest GPX and highest T-bars were observed in control group as compared to the supplemented groups. Vitamin C content in liver. Vitamin C content in liver increased in a dose-dependent manner with vitamin C dose, and all supplemented groups were significantly different from each other except 50 and 100 milligram per kilogram level that were non-significant to each other. Survival rate against Aeromonas hydrophila. Survival rate of supplemented groups were significantly higher than the control group. However, a significant decrease was observed at 800 milligram per kilogram level. Optimum dietary vitamin C requirement based on the broken line regression model of weight gain percent. The optimum dietary vitamin C requirement of silver carp fingerlings was estimated to 50.25 milligram per kilogram. Conclusion and suggestions. Conclusion: the dietary vitamin C significantly enhanced the growth performance of silver carp fingerlings by improving the proximate composition, hematology, antioxidant enzyme activities, and survival rate against pathogenic strain. The optimum dietary vitamin C requirement of silver carp fingerlings was 250.25 milligram per kilogram. Suggestions: a local farmer can increase its fish production by using the diet supplemented with optimum level of vitamin C. This will also help the fish to mitigate the various stress produced during aquaculture practices. This research should be helpful for both small and large-scale aquaculture by improving the growth and survival rate of silver carp fingerlings. Thank you.
Thank you, Ms. Mavish, for presenting your research work at ICAZ. Now I would like to call Ms. Fatima Khalid from Anhui, China, and she will deliver her talk on production of industrially important enzyme protease from Bacillus willisensis Y1 strain isolated from the manure of piglets. Please, Ms. Fatima Khalid.
Hello everyone, this is Fatima Khalid. I'm MS student at School of Life Sciences, Anhui Agricultural University, Hefei, China. Today the topic of my presentation is production of industrially important enzyme protease from Bacillus willisensis Y1 strain isolated from the manure of piglets. So moving forward, first of all, I will give you the brief introduction of the enzyme and this bacterial species. So, as we all know that enzymes are most efficient biocatalysts, they provide numerous benefits as compared to traditional chemical catalysts. So, they are considered as environmental friendly and cost alternative to the chemical catalysts used in the industry. The enzymes from Bacillus phalanensis have been verified to be the key substance to inhibit the parasitic harmful fungus and bacteria both in plants and animals. So, as a novel biocontrol bacterium, Bacillus has been widely used as a biological control agent in agricultural field due to its strong ability to suppress other pathogenic bacteria. Here you can see a 3D model of protease enzyme. Next, what are the objectives of this study? So, some series of the known proteases aren't enough to accomplish the industrial demand of enzymes. So, the study of protease production from Bacillus phalanensis has extremely important industrially value. In order to industrialize this enzyme, this study aimed to optimize different fermentation conditions for protease isolated from the manure of piglets. Moving on, the enzyme properties were investigated and its genes were cloned. So, what are the materials and methods that were used in this study? First of all, what we did was we prepared an LP culture medium. All the equipments used in the experiment were sterilized, and all the experimental steps were performed quickly so to prevent contamination. And the isolated bacterial colony was subjected to morphological analysis by smear preparation, gram staining, followed by microscopic examination. And then genomic DNA was extracted using a bacterial DNA extraction kit according to the manual followed by the given on the extraction kit. And then application of the 16S ribosomal gene was done by using primers. Bacterial culture in the fermentation medium was continuously centrifuged to obtain a crude enzyme. And the activity of protease was determined by Folin phenol method. And optimization was done for fermentation of different parameters such as temperature, time, pH, liquid load, carbon to nitrogen ratio. And then orthogonal test for optimization of fermentation conditions was carried out, and each factor was designed with 3 experimental levels to avoid any mistake. Here is a graphical representation of the method. So we used solid state fermentation for the optimization of different fermentation conditions like temperature, time, pH, liquid load, carbon to nitrogen ratio, so on. And then the enzyme production was done. So what we conclude from this study? A strain producing protease identified as Bacillus phalanensis Y1 was isolated from the manure of piglets. High yield of remarkably important biocatalyst protease was obtained by optimizing different conditions by using solid state fermentation, and it is considered industrially important. So there is only a little research on this strain to be used as probiotic in animal feed as for the replacement of antibiotics for which our bodies has developed resistance. And We hope that in the near future, research on the replacement of antibiotics by Bacillus thuringiensis in animal feed will be reported by our laboratory. Thank you for your attention.
Thank you, Ms. Fatma Khalid, for such an interesting talk. Now I request Ms. Umme Hani from Fatma Jinnah Women University to present her talk on redesigning the conservation efforts for endangered mammals in Pakistan considering climate change. Please, Ms. Umme Hani.
I'm Umehani, and I'm here to present my research article, which is on redesigning the conservation effort for endangered mammals in Pakistan considering climate change. I am from Fatma Jinnah Women University and from Department of Environmental Sciences. The introduction to my this review article is basically related to climate change, as it is impacting the global ecosystem through fluctuations in temperature and precipitation. Climate change, like all other aspects, is also affecting biodiversity. Hence, before the conservation efforts for biodiversity, it is very important to consider the impacts of climate change on it. As IPCC has projected a rise of about 0.15 to 0.3 degrees Celsius in temperature in this century, by the end of the century, and climate change is anticipated to influence the maximum part of ecosystem and levels with a more harsh impact on them as the time will progress. Next, I'll discuss the status of mammals in Pakistan and the major threats to their existence. The major impacts basically on mammals are on their physiology, their biology, and their dispersal, more specifically due to climate change. Their habitat is going to disperse and their range is going to shift, due to which their ecological niche is going to modify in the future due to impacts of climate change. When we talk about the status of mammals in Pakistan, we come to know that most of the mammals in Pakistan are in the category of endangered, data deficient, and near endangered categories, while some of them are critically endangered. So it is need of the hour to think about their conservation. But before thinking about their conservation, doing their conservation, it is very important to anticipate all the major threats to them. So for this purpose, the methodology which I have designed in my review article was that I selected the 5 major projects of mammals related to conservation of mammals in Pakistan. And after selecting them, I thoroughly revisited the strategies these projects have considered. Moreover, after observing these strategies, I analyzed them for climate change, the most specific impacts of climate change on the particular species on which the projects were. And after considering these impacts, I have modified the projects according to the anticipated threats. The strategy development was based on the assessment of ecosystem, the conservation of species concerns, the major project goal, the threats to them, and the measures of progress. After my methodology, I opted for results and discussion, my presentation. The first important result of my article was that I considered a project for the conservation of musk deer. The threats which I have identified to musk deer were illegal hunting and poaching for their musk, the deforestation, overgrazing, and global warming. The hypothesis of change for this species is that deforestation has resulting in the fragmentation of the habitat of this species due to increased temperature and recession. The range of musk deer is going to shift to higher elevation, which means that its habitat is going to modify.
So under these changed circumstances, the mitigation strategy which I have devised is resistance. Under resistance, the following measures should be taken: establishment of corridors for safe movement, enhancement of forest cover, and shifting of local population away from core areas of must clear. The second important conservation project which I have selected was related to Indian pangolin, because the major threats to the Indian pangolin is about the killing of Indian pangolin for its scale and body parts, its illegal hunting and poaching. Under climate change, there will be a lot of more specific impact on the habitat of this species, as Indian pangolin established two types of burrows, the resting and feeding burrows. And these burrows usually lie away from human settlements. Under this current scenario of urbanization, the habitat of pangolin is going to shift towards more towards the areas which are not very much inhabited. So the temperature and humidity of burrows is also a very important factor, which is going to change under this climate change scenario. Hence, devised a mitigation strategy for this species resilience. Resilience means improvement for the habitat, improvement of the habitat condition by placing more boulders in its range. Resting boulders should be associated with the buttresses of the roots of several trees, and habitat-specific measures should be taken. The other project which I selected for the conservation of snow leopard, the major threats to the snow leopard is its hunting for its body parts, its illegal poaching, and killing by herders. The hypothesis of change which I have devised is that under climate change, the snow leopard is moving to higher elevations, and due to its movement to higher elevation and encroachment of human being to its range, is shrinking its habitat and its area where it moves and breed. So the species is going to breed less and the population will decline, not only in Pakistan but also in other associated countries, because the snow leopard is a range species, and it moves across its range in 12 different countries. Its range is spread over, and the devised mitigation strategy for this species to protect it from the threats of climate change is resilience, to ensure the access to a secure prey base of this species so that it does not compete with the human of the area, and acquisition of a better understanding of snow leopard biology and disease and genetics, and creating a condition for human population to adapt better to the impacts of climate change in the snow leopard range. Other measure is to focus on minimizing the current threats such as poaching, killing, and habitat degradation. Next species is common leopard. Common leopard is threatened by human-leopard conflict, retaliatory killings, and encroachments. Moreover, its natural prey is also decreasing, and trade of its body part is also common. So the hypothesis of climate change is that the leopards are resilient to direct impact of climate change, but due to increasing temperatures and famines, a lot of indirect impacts will happen to this species, due to which it will move towards, for food and for other resources, it will move towards the residential areas, due to which the human leopard conflict will further increase and retaliatory killings will result in decrease in its population. So the devised mitigation strategy is resistance. Resistance include the following measures: the protection of refugia and creating corridors to allow the species movement, expansion of protected areas to include the migration corridors and seasonal feeding areas, and improving the connectivity of the habitats. The last conservation project which I selected for this review article was about Indus Dolphin. The major threats to Indus dolphins are poaching, accidental killings, fishing, net entanglement, use of chemical and fertilizers. So the hypothesis of change which I have devised is that the growing temperature and unpredictable rainfall are leading to fast deterioration of river flow, and that is designed to hit the dolphin hard. So it is very important to compete and manage the increasing temperature, which is going to increase more and more by the end of this century, and to control the illegal illnesses, new illnesses, and illegal hunting and fishing of this species, so that its population can survive, because as it is an endemic species to Indus River of Pakistan. The major devised mitigation strategy measures which I have designed for this specific species, that it's about transformation of its habitat. It involves the proactively translocation of the species beyond the current range limit and reduce the non-climate stressors on natural areas in the ecosystem, and translocation or assisted dispersal of the species at risk. That's all about my presentation. At conclusion, I want to say that understanding and awareness regarding the complex interaction of climate change with the mammal species is a dire need of ours. To help such species survive and maintain healthy ecosystem is now our duty to consider climate change. It's not only a theoretical or hypothetical thing. It's happening, it's impacting all life forms, and specifically the mammals, not only in Pakistan but across the world. So it's our duty and the duty of our conservation organizations to consider climate change before designing any conservation strategy. These are some references from where I have collected my data. Thank you so much for your attention and your time, and for listening.
Thank you, Ms. Omehani, for such an informative presentation. Now I request Mr. Mohammad Tariq from University of Lahore to present his talk on ecological modeling of red-vented bulbul Cainotus kefir by GIS application in district Niawali, Punjab, Pakistan. Please, Mohammad Tariq.
My name is Tariq Khan and I am a student of University of Lahore Subcampus Sargodha. My topic of presentation is ecological modeling of red-vented bulbul by GIS application in district Niawali, Punjab, Pakistan. Introduction. Red-vented bulbul is a medium-sized, long-tailed serine bird having short crest on head, which gives the head a squarish appearance. The body is dark brown with a scaly pattern, while the head is darker or black. The rump is white, while the vent is red. The black tail is tipped in white. The Ficunidia family is the largest group of passerine birds with about 138 species, out of which 5 species are recorded to happen in Pakistan. In Pakistan, it is widely distributed throughout the Indus Plain and cultivated areas of all provinces except Balochistan or any desert area. The red-vented bulbul is recorded in the IUCN Red List of bird species as the least of concern. The Geographical Information System is a computer system for capturing, storing, checking, and displaying data related to position on our surface. GIS can show different kind of data on one map, such as road, river, lake, vegetation. Main function of a GIS is a visual representation of data. GIS provide a database for the geospatial data to be stored in and then represented visually in a map format. Objective of our study to investigate the spatial distribution of red-vented bulbul using GIS technique to assess the information of nesting and breeding biology of red-vented bulbul. Research methodology. The flow examination will be directed in district Niawali, having 8 destinations to check the ecology of red-vented bulbul by utilizing GIS-based method. All GIS work will be performed by utilizing RMAP 10.7. Study area. The current examination will be coordinated in Mianwali district. Mianwali is situated in the northwest of Punjab, Pakistan. Mianwali district covers an area of 5,840 square kilometers. Map showing the study area, yellow line showing the district boundary, red line showing the tehsil boundary, and blue triangle showing the study sites. We have total 8 sites which are named Chikdala, Samas, Namal Lake, Musakhel, Abakhel, Bitiya, Gulmiri, and Chidru. Method of study. The fixed line transect 1.5 km was used in all of the 8 attempts having red-vented bulbul population. A group of 2 knowledgeable persons, local farmer and resident, along with author, work together along the transect line at the morning and evening session during different months of year 2021 and record the sighted and calling red-vented bulbul. Statistical analysis with the help of MS Excel Office 2019 was used for the statistical analysis such as mean percentage and standard deviation to analyze the data. Results. General distribution. The data accumulated on the distribution of red-vented bulbul from 8 destinations analyzed in the Mianwali. The red-vented bulbul species were equally distributed in the examination zone. The red-vented bulbul species gave it appearance in 7 attempts. The distribution of red-vented bulbul in district Mianwali can be seen in figure 1. Map showing the distribution of red-vented bulbul, in which dark red color showing the district boundary and red color showing the tehsil boundary, in which red color showing the study sites and the green star showing the nest, and these red arrow showing the circulation of bird in a zoo. Courtship display. The first courtship display of red-vented bulbul seen in mid-February 2021 nest size selection. The first nest was seen on late February 2021. Complete 42 nests have been seen during the investigation. Out of 42, 9 nests have been seen in Chikdala, 5 in Sawan, 7 in Musakhel, 4 in Namalek, 6 in Abakhel, 5 in Bitiya, 6 in Chidru. No nest is found in Gulmiri. Map showing the study site Chikdala. In this site, we have total 9 nests have been observed and identified. Map showing the study site Samas, in which total 5 nests have been identified and observed. Map showing the study site Musakhel, in which total 7 nests have been identified and observed. Map showing the study site Namalek, in which 4 nests have been identified and observed. Study site map showing the study site Abakhel, in which total 6 nests have been identified and observed. Study site showing Bitiya, in which total 5 nests have been identified and observed. Map showing the study site Chidru, in which total 6 nests were identified and observed. Map showing the study site Gulmiri, in which no nests have been found. The reason behind the absence of nests is due to high human populated area, and there was no preferable tree for the construction of nest construction. It was seen that the red-vented bulbul make nest from late February to late July 2021. Red-vented bulbul mainly utilize plant material for nest construction like plant root, twig, plastic fiber, grasset, and spider webs. Total nests were noticed 42, out of which 7 were inactive and 35 were active. Red-vented bulbul prefer to make nest on Gawa, Kokanbe, Sanatha, Tali, date palm, Largai, Chambeli, white mulberry, sweet orange. Prefer height of nest is 1 to 4 meter. The size of red-vented bulbul is 1 to 4x. Incubation period of red-vented bulbul egg varies from 11 to 14 days. Fledgling periods were recorded as 13 to 16 days. Egg of red-vented bulbul are oval shaped and the color is pale pinkish with the spot of darker red, more dense at the bend. Egg and nestling loss of red-vented bulbul. We have total 95 number of egg observed, in which 8 number of egg are infertile, 6 are predated, and 3 were damaged due to climatic conditions. So the hatching success of our study is 82%. Number of nestling, we have total 78 number of nestling which we observed, in which 8 number of nestling are predated, 2 were died due to weather condition. So the fledgling success of our study is 87%. Conclusion. From the current study, it can be concluded that this species is widely distributed among the study area. Some variation arises due to variation in habitat available, season, and climatic condition. Total 42 nests were identified during study. Success rate of hatchling and fledgling was 82 and 87% respectively. Most of egg were lost due to predation and harsh environmental conditions. Highest number of eggs were observed in region Chikdala, followed by Musafir. No egg were found in region Gulmiri. Eggs were constructed by the plant material such as twig and rootlet grasses. Preferred plant position was fork tree under the dense canopy. This research will help to establish future conservation strategies. Moreover, GIS mapping is one of the most effective techniques for analyzing the spatial distribution of birds. Thank you so much.
Thank you, Mr. Muhammad Tariq, for sharing your research work at ICAZ. Now I would like to call Ms. Sidra Bano from University of Agriculture, Faisalabad. She will deliver her talk on ecological impact of benzophenone type UV filter on the catalase and peroxidase activity in grass carp, Stenophryngodon adela. Please, Ms. Sidra Bano.
First of all, I am very thankful to the organizing committee of ICAZ-2021 for providing me an opportunity to share my research work with scientific community. My name is Sidra Bano, and I am from the Zoology, Wildlife and Fisheries Department of University of Agriculture, Faisalabad. My research topic is ecological impact of benzophenone type UV filter on the catalase and peroxidase activity in the grass carp, Stenophryngodon adela. Introductions. UV rays have adverse health effects and can cause cancer and eczema-like disease. To protect the skin from the harms of UV rays, various cosmetic products are used. These products contain UV filters. UV filters are very large and heterogeneous group of chemicals used as a cosmetic ingredients in several personal care products to protect the skin from the harms of chronic and acute impact of UV rays A and UVB. UVA wavelength range from 320 to 400 nanometer and UVB rays wavelength range from 280 to 320 nanometer. On the basis of their chemical composition, UV filters are either organic and inorganic. But most widely used UV filters belong to the organic aromatic compounds family, and benzophenone 2 is one of them. Benzophenone 2,2,4 tetrahydroxybenzophenone is an organic UV filter used in various personal care products such as soaps, aftershave lotions, lipsticks, shampoos, and sunscreens. Along with this, they are also used in pharmaceutical industry and in plastic industries. Now the question arises how these contaminants are added into the water. Due to the recreational activities and wastewater treatment plants, these contaminants enter into the waters. According to an assumption, about 10,000 tons of UV filters are produced annually to meet the demand of the global market. They are considered as an emergent pollutant of concern by the environmental protection agencies. In ancient time, it was suggested that UV filters have adverse health effects related to the skin, but recent research proved that they can cause systematic toxicity, as they enter into the blood and circulate throughout the body and can cause systematic toxicity, such as they can generate reactive oxygen species such as the hydroxyl radical and superoxide radical. Under natural conditions, all living organisms have a defensive system to protect them from the harms of reactive oxygen species. These defensive systems include enzymatic antioxidant and defense system and non-enzymatic antioxidant defense system. These enzymes which are perform antioxidant activities are superoxide dismutase, catalase, glutathione peroxidase, peroxidase, glutathione reductase, glutathione esterase, while the non-enzymatic non-enzymatic defense system include the vitamin A and the C. Actually, when the imbalance between the reactive oxygen species and this antioxidant enzyme activity occurred, then the oxidative stress resulted, and the oxidative stress is a very damaged conditions for living organism, as it can cause lipid peroxidations and can also damage the protein, as it causes protein carbonylation and can damage the DNA. And a lot of the production of the reactive oxygen species can cause canceration and the death. The aims and the objective. The aims and the objective of my research were to check the acute toxicity 96-hour LC50 and lethal concentration of benzoquinone 2 in grass carp, and to determine the effect of sublethal concentration of benzoquinone 2 on catalase and peroxidase activity in grass carp. Materials and method. First of all, I chosen the grass carp as an experimental organism due to its various ecological importance. Grass carp is acts as a biological control of herbs in fish farms, and it is also a good source of protein. So I chosen the grass carp as an experimental organism, and I purchased them from the fish seed hatchery Satyana Road, Faisalabad. Then I transferred them to the fisheries research farm of University of Agriculture, Faisalabad, and where they were placed in a cemented tanks for at least 2 weeks for the acclimatizations. And during this acclimatization period, they were fed with feed, which was 4% of their live body weight. After that, they were transferred to the aquariums of 50 liters, and 10 fishes were allocated in respective aquariums. Chemical preparation. Stock solution was prepared by the using dimethyl sulfide oxide as a solvent, while the working solution was prepared by the dilution of the stock solution with distilled water. Assessment of 96-hour LC50. For assessing the 96-hour LC50, different concentration of benzoquinone 2 were given to fish, and their mortality rates were checked. And these concentrations were gradually increased to avoid the risk of stress in them. And probability analysis method was used to determine the 96-hour LC50 concentration of benzoquinone 2 for grass carp, tinopen, godon, idilla. After assessing the 96-hour LC50 of benzoquinone 2 for grass carp, then I divided the fish into 3 groups, and each group contained 20 fishes, and group 1 named them group 1, group 2, and the group 3. And different concentrations of benzoquinone 2 were given to them, such as 1/7 LC50 concentration of benzoquinone was given to group 1, 1/5 LC50 concentration of benzoquinone 2 was given to group 2, and 1/2 LC50 concentration of benzoquinone was given to group 3. After fish sampling, fish dissection was done, and major organs such as the liver, gills, muscles, and hearts were extracted and stored in refrigerator for further antioxidant enzymatic activity studies. Enzyme assay. Peroxidase enzyme assay. The peroxidase enzyme activity was calculated by the method of the Saberi et al. 1995, and the basic principle of this method was that the antioxidant ability of peroxidase enzyme was measured by its ability to reduce the hydrogen peroxide per minute. Catalase enzyme assay. Catalase activity was measured by the method of Chance and Mahle, and the basic principle was that catalase activity increased are measured by measuring its stability to reduce the hydrogen peroxide per minute. Different statistical methods were used, such as a probability analysis method was used to determine the 96-hour LC50 ANOVA, and correlation was used to determine the relationship between different physicochemical parameters, and Tuckey test was used to determine the comparison between mean values. These are the results figure. One shows the catalase activity in different organs of grass carp during acute exposure of benzoquinone 2. As the fish were exposed to different concentration of the benzoquinone 2, such as 1/7 LC50 concentration of benzoquinone 2, 1/5 LC50 concentration of benzoquinone 2, and 1/2 LC50 concentration of benzoquinone 2, their catalase activity was increased. This is due to the increase in the production of reactive oxygen species, and so the catalase activity was increased, and high catalase activity was observed in liver than other organ, because liver is the major site of detoxification. And as the exposure duration was increased, the catalase activity was further increased. To show the peroxidase activity in different organs of grass carp during acute exposure of benzophenone 2, peroxidase activity increased with as the concentration of benzophenone 2 increased, as such as a high activity was observed in fishes that was treated with 1/2 SC50 concentrations, and because it is the highest concentrations and high activity was observed in liver, then gills, kidney, muscles, and the heart. And as the duration of the exposure increased, then the peroxidase activity was also increased, such as the high peroxidase activity was observed after 96 hours. Conclusion: exposure of the benzophenone 2 to the grass carp causes the changes in antioxidant enzyme activity, and these changes were highly significant, as the p-value was 0.01, and antioxidant activity was higher in liver than other organs, because liver is the major site of detoxifications, and there are no subsequent changes in antioxidant enzyme activity in the control group. Overall, catalase and peroxidase activity show increasing trend in the following orders: liver, gills, kidney, muscles, and high activity was observed in liver, then gills, then kidney, and muscles. It can be observed that both catalase and peroxidase activity increased with period in the following order: high catalase and peroxidase activity was observed after 96 hours, then 72, then 48, and least activity was observed after 24 hours. And this activity was increased because the reactive oxygen species production increased as the exposure duration was increased. These are the references.
Thank you, Mr. Daf, for presenting an interesting research work. Now I request Dr. Muhammad Kabir from University of Sargodha to present his talk on environmental pollution and its nature-based solution for sustainable habitat of living organisms. Please, Dr. Muhammad Kabir.
Bismillah Rahman Rahim. Respected chair, co-chair, and audience, Assalamu alaikum. I am Dr. Muhammad Kabir, working as an assistant professor and in charge at Department of Biological Sciences, University of Sargodha, Subcampus Bakhar, Punjab, Pakistan. First of all, I pay my thanks to the organizing committee of the 4th International Conference on Applied Zoology 2021, who gave me a chance of oral presentation in this conference. Topic of my presentation is the environmental pollution and its nature-based solution for sustainable habitat of living organisms. As we know that research starts with the problem and ends with the solution, so the presentation layout includes the problem, that is the pollution, and its solution, that is the plantation. Importance of tree for sustainable habitat, pollution, pollutant types, and issue, plantation campaign, solution for pollution, environmental calendar, conclusions, and recommendations. As we know that plants are producer on the surface of earth, they carry out the process of photosynthesis, and as a result of photosynthesis, they produce the photoassimilates or photosynthetic product, that is the glucose. In this process, they also take up the carbon dioxide from the environment and give out oxygen for the existence of living organism on the surface of earth. So the importance of the tree on the surface of earth cannot be denied. Muscosol says a tree that lives for 50 years generates $US worth of oxygen, recycles $US worth of the soil fertility, facilitates $US worth of soil erosion control, creates $US worth of the air pollution control, and provides a huge quantity of $US worth of shelter for insect, birds, and animals. Besides, it provides flower, fruit, and seed. Our net loss is worth more than $US 57,381 when one tree fall are affected by environmental pollution. Therefore, a humble request is that think before you cut a tree. What is the pollution, pollutant, their types, and issue? Pollution, the undesirable change in physical, chemical, and biological characteristics of the air, water, and soil that may harmfully affect to the living organism is known as the pollution. Pollutant, the substances which cause the pollutions are known as the pollutant, or simply we can say the residue of things we make, use, and throw away are known as the pollutant. As we know that environment is consist of the three major components, that is air, water, and soil, and all these components of the environments are degrading due to the anthropogenic activities, mainly due to the industrial activities and the transportation activities. So the pollution or pollutant types includes the air pollution, water pollution, soil pollution, noise pollution, thermal pollution, chemical pollution, and etc. What is the issue? The issue is that we use the resources extensively and then are not responsible for the consequences. Man can go up to any extent for the utilization of the natural resources without considering the importance of environment. Our industrial and transportation activity are on large scale, but I will focus to the industrial activities. It is our common practice that most of the industrial wastes are dumped outside the boundary wall, and by their burning process, different types of the pollutants are being discharged into the all components of the environment, including air, water, and soil. Second activity which is being carried out by the human on the surface of Earth is deforestation. We are establishing the industrial state industrial units on one hand, and on the other hand, we are carrying out the deforestation activity. One thing is the cutting of the tree, and another is that these cutted trees are burned, as a result of which different types of the pollutant are being released into the environment. So what is the solution for pollution? As I already mentioned that plants are the producer on the surface of the earth. Plants act as the natural lung. So they also provide the nutrition to the living organism. They also provide oxygen for existence of life on the surface of earth. It means that plants are very important in order to maintain the natural habitat of the living organisms. If we will carry out the plantations activity on the large scale, it means that we are giving the more sustainable environment to our next coming generation. And if we are carrying out the deforestation activity on the large scale, it means that we are not sincere about our environmental scenario for the next coming generation. So plants act as the natural lungs. Plant a tree and get oxygen for free. Plants are the natural purifier of the environment, as they give out the oxygen and take up the carbon dioxide during the process of the photosynthesis. And this increasing concentration of the carbon dioxide is mainly responsible to increase the temperature on the surface of Earth, for the global warming, for the ozone layer depletion. So different gases which are being produced from the transportation activity and industrial activities are mainly responsible to pollute all components of the environment. It is our duty to save the environment beauty. Let us go green to get globe clean. Green revolution is the best solution to arrest the pollution. As moving towards the solution for pollution, in our sub-campus, entrance gates are also made by different types of the trees, by different types of the plants, creepers, so that a student can get their entry into the campus after knowing the importance of the trees. In front of our building, different plants are also potted in the earthen pots to keep the environment clean and green. Our faculty member, our students and university staff also involved in plantations activity, not only within the campus but even in the district level, with in collaborations with the Deputy Commissioner Office, with the Regional Forest Officer, with Environmental Protection Agency, in order to keep the environment clean and green. Our worthy director is planting tree in our sub-campus. Our student, in collaboration with the Pakistan Army, also planted different trees in our sub-campus. The issue is that we are not focusing to the importance of the environment for the proper habitats of the living organisms. As we know that we celebrate only one day annually, that is our birthday, by cutting the cake, and we are not familiar with the different events of the environmental calendar. We should focus to some important events of the environmental calendar if we are environmentally friendly. The environmental calendar most important events includes the World Environmental Day, that is celebrated on the 5th of June. World Population Day is celebrated on the 11th July. Ozone Week from 16 to 23rd September, and Green Consumer Day on the 28th September. Similarly, World Habitat Day is celebrated on the 3rd of October. International Day for Natural Disaster on 13th of October. And similarly, the last one event is the International Plantation Day, which is celebrated on the 18th of August. So we should focus to the plantations activity by keeping in view the environmental calendars. What is the conclusions? If haphazard population growth and construction of the new industrial structure goes on without realizing the importance of the tree, then probably there would be more environmental changes in the near future. And these environmental changes are harmfully affecting to the habitat of the living organisms on the surface of Earth. On the basis of these conclusions, or study, I would suggest the recommendation that be a part of the solution, not pollution. And I also have a request to all the participants of the conference that each one must plant one and join the green revolution and stop the pollution. Thank you for your attention.
Thank you, Dr. Kabir, for such a nice presentation. Now I request Ms. Laiba Amin to moderate third session, which will include talk from the fields of parasitology, toxicology, and pathology. Please, Ms. Laiba Amin.